Hi, everyone. This is Jason Key at SBGrid at Harvard Medical School in Boston. So thanks for joining us today. Today, I'd like to introduce John Stone, who's joining us from the University of Illinois, Urbana-Champaign, and Abhishek Singharoy from Arizona State. They're going to tell us about VMD. So John and Abhi, are you there?
Yes.
All right, great. So John's going to kick it off, and then we'll transition over to Abhi halfway through. If anyone has any questions, feel free to send them to me by chat, and I can pass them on. If there's something that's sort of particular to a slide, we can interrupt and just clarify. Otherwise, I'll probably save them toward the end. And then, yeah, with that, John, go ahead.
Thank you. So we got your systems check slide there. We ready to go?
Good. We're ready to go.
All right. So, good morning. My name is John Stone. I'm a senior research programmer at the University of Illinois. I've been the main developer of VMD for the last 22 years. Today, we're going to talk about VMD and its use for preparation and analysis and visualization of molecular dynamics simulations. And for today's talk, we're going to focus a little bit more on what I refer to as hybrid simulations, or simulations that are not straight MD, but maybe that relate to experimental imaging, structure refinement, things like that. So VMD has been designed, if you look at various molecular visualization tools in the field, VMD began just as a visualization tool, and its key differentiator was that it was organized around molecular dynamics, so time-varying structures from the very, very beginning. And over the years, VMD has evolved to incorporate built-in analysis features and eventually GPU acceleration, and now parallel analysis on supercomputers. And the diversity of structure information it works with has grown. It began just with all-atom atomic structures, and now we are able to work with a wide variety of cryo-EM and cryo-ET density maps, volume data of various kinds, electrostatic potential maps, quantum chemistry data, and sequence information. And so VMD works with all these different types of data. If you look at how VMD is used in practice today, it acts sort of as an in-between, or between the experimental structure information, different modalities of structure information, and it provides the platform and some of the tools to prepare the structures that are used in a molecular dynamics simulation. It works with all the major MD packages, and it also works with other types of simulations, lattice cell simulations, various types of Brownian dynamics simulations. And as the program has grown, the different hardware platforms it supports have grown as well. So it began life as a VR application that ran in the cave, evolved to work on desktop and laptop computers, and today it runs on everything from a laptop all the way up to cloud nodes and supercomputers. And some of the direction we've been growing the program in the last several years has been to improve its features specifically for hybrid modeling or data-driven simulations. These are particularly things that relate to structure refinement with molecular dynamics flexible fitting, other modalities, and not just things that we develop here, but also tools that others outside in the research community have developed. VMD is very extensible. It has built-in scripting languages. It incorporates support for both TCL and Python 3. We are expanding the range of different tools it provides for these purposes, and we don't view VMD as a one-stop shop for everything. So it's important to us to link to the other tools that are developed in the community, and we want to make our software and the components that make VMD special available to others to use as well. So we've developed many versions of VMD over the time I've been in charge, going back to 1998. Our user community has grown tremendously. Today, VMD is cited by a research paper once every 2.6 hours, which is mind-boggling to me compared to when it first began. Some of the things that we've been working on just in the last 2 years have been extending the program's support for advanced rendering. So I'll talk about interactive ray tracing in a little bit. We had to do a major update to support Python 3. We're working on improving its interactions with different secondary structure calculations. It has had a built-in tool called Stride that was developed externally, and now we link VMD to tools like XDSSP, and we're working on developing GPU-accelerated tools for secondary structure calculations. We have a lot of investment in GPU algorithms for processing volumetric data like density maps, things like this. And we have some really unique features for hybrid quantum chemistry molecular mechanics simulations. And so I'll show you, and I think Abhi has several nice examples of those. One of the key things that we did differently about VMD from the very beginning is that it had a built-in scripting language. And that, in the early days, enabled people to customize or write special scripts for their particular analysis capabilities they wanted. And this evolved to the point that people could write graphical user interfaces. And we now have formalized this mechanism, and it is pervasive throughout VMD, and allows anyone to expand the program's support for different file formats, to develop specialized tools to do different kinds of analysis. And so just to give you a few examples of those, I'll show you in a few slides some of the ways those are implemented. So one of the things that VMD has been known for a long time is its high-end visualization features. Up to the late 2000s, the main way that we generated images was with a technique called rasterization. That's what OpenGL provides. And going forward, one of the state-of-the-art techniques that's possible today is called ray tracing. VMD has had support for ray tracing for the entire time it's existed. However, in the early days, it was a batch mode process that was very slow, and it might take even an hour to generate an image. Today, with state-of-the-art techniques, ray tracing can be done interactively at 20 frames a second on a desktop computer with a GPU or a recent CPU. And that means that we can use very high fidelity lighting and shading techniques that weren't previously possible. So just to give you an example, if you were to use a conventional molecular visualization tool written with classic OpenGL, and you were visualizing a virus capsid and you cut the capsid in half, the image on the left is the sort of thing you get with simplistic direct lighting that a lot of the prevalent molecular visualization tools provide. On the right hand, we have an image that shows what's called ambient occlusion lighting, which uses complex simulation of light scattering, and it approximates what you'd get if you took a photograph with a broad light source, much like if you take a portrait photo in a photography studio. This is more the sort of shadows the photographer is trying to get. And you can see there's a huge difference in the salient details that you can see in the image on the left versus the image on the right. Perhaps the most important thing about ambient occlusion lighting is the fact that the scientist who's generating these molecular graphics doesn't have to adjust anything to get that outcome. This is sort of an automatic mechanism. So by using the more advanced lighting model, the computer is working harder to give us a better image that's easier to interpret. You can see pockets, pores, cavities, all of the key structural features that are important to see in a complex 3D molecular structure. And another thing that's nice about ray tracing is it gracefully handles things like spheres and cylinders, which compose a lot of the geometry we draw with all atom models. And ray tracing also works very well for volume rendering, for viewing things like density maps. And so one of the great advancements of the last year has been support for state-of-the-art GPUs. Some of the latest GPUs made by NVIDIA now have a feature called RTX. It's used by video games and other applications also, and its purpose is to accelerate ray tracing in hardware to give you a significant speed bump or speed increase over what you had in previous hardware generations. So here are two nice examples. These are very large photosynthetic organelle models. Abhi will probably show you several more pictures of this. This part of the structure you're seeing on the left is a scene with about 10 million atoms in the outer part of the structure, rendering a 4K by 4K image with an older GPU with a normalized performance. I think this is something that renders in about 30 seconds with very high quality and all the dials maximized for a manuscript figure. Now we can do that in 1/8 the time. So the new hardware performs 8 times faster. So the code in VMD was updated to exploit the state-of-the-art hardware. And so the benefit to the user is things that were previously a non-interactive scene. For example, the one on the right, I can tell you when this was from a cover image we made for a journal. When we first made that image, it took 3 GPUs, and it was barely interactive. With 3 GPUs, we'd get about 3 frames per second. And now we can do the same thing with a single RTX GPU, and the performance is about 24 frames a second 3 years later and 2 hardware generations later. So that's very exciting. So that's really the benefit of hardware-accelerated ray tracing. Abhi will show you a lot of other cool things that are rendered in that way. VMD was just used to make a planetarium video that's going to be shown all over the world. That's premiered in Germany, and this is the first time VMD's built-in render has ever been used for production rendering for a movie, and it's very exciting. So that's another thing that the ray tracing techniques allowed us to do, is we could generate 180-degree field of view projections for planetariums. And now, more recently, the state-of-the-art versions of VMD can generate VR movies that can be shown on YouTube. So you can display them with Google Cardboard, or you can, even with a plain web browser, you can rotate the movie and view it in all different directions. That gives you the freedom to turn your head around and look at different things. And you retain all the same lighting and shading features you had in a conventional molecular graphics movie, but now you can explore and you can make omnidirectional movies of your simulations. Going forward, we've done some groundwork to support ray tracing in VR headsets, and we expect continuing work there to enable this on some of the state-of-the-art headsets like the HTC Vive going forward. So it's a very exciting area of development. So as I talked about, aside from the visualization, which is of course a major part of what we do in VMD, the other things are all these tools that people use to do structure preparation, to do analysis. This is actually a slightly out-of-date list, but it exemplifies the diversity of different tools that VMD contains. And another thing I wanted to point out is that these tools aren't all developed at Illinois. A lot of them are actually developed by our collaborators all over the world. So the ones that are shown in green are developed by other people, and they're contributed to VMD, and we make them available to the user community as well. So you can take a tool like VMD and extend it to do new things just by writing some scripts. You can add your own graphical user interfaces and your own analysis calculations. So some of these things, as an example, we have a new molecule building tool called Molefacture. There was a previous version of Molefacture that was I guess you'd say it was limited to very small structures of 100 atoms or less. The new version has a completely redesigned graphical interface, and it has design templates and an efficient workflow for building proteins and more complex molecules. It's far, far easier to use than what we had before. And it now has linkage to tools to do things like minimizing the structure. It can also get force field parameters. So since a lot of the usage of VMD is to prepare molecular dynamics simulations, it's important that when we build novel structures and things like this, we also have tools to obtain the force field parameters that allow them to be simulated in a tool like NAMD or AMBER or GROMACS. We have another tool called QwikMD, which automates the process of preparing a simulation. So it can take both raw structures out of the protein data bank or from something like Molefacture, and it can add solvent and ions and all the other different tasks that a user might have to perform, get a simulation ready. And it has a lot of different automated features to assist the scientist in making important decisions about how to prepare that structure. It doesn't really, it's not a black box. It doesn't take control away from the scientist. We designed this tool really to leave all the key decision points in the hands of the scientist, but inform them of where there are troubles with their structure and to ask them what remediation should be taken. And another thing about the tool is that it's designed for reproducibility. It logs all the different steps that are performed, and those are both machine readable and human readable, so that you can hand a simulation that you've prepared with QwikMD to one of your colleagues, and they should be able to reproduce everything that you did. VMD has some new features for analyzing large biomolecular containers, things like virus capsids or other subcellular organelles, things where you have membranes or other enclosed spaces. The tool can analyze the different parts of the structure and classify things as interior, exterior, or surface of the container. And these analyses can be used to do other complicated bookkeeping over the course of a simulation to determine, for example, how many ions move in or out of a virus capsid over the course of a certain period of time. Lots of things like this. And this is a sort of a building block you can use to do much more sophisticated analysis. And I think Abhi will tell you a little bit about that. Some of the things we're working on presently are to extend VMD to support next generation hybrid modeling efforts. So you may be aware that the PDB has an integrative hybrid modeling project, and there is a new extended version of the PDB dictionary that allows you to store a wide variety of structure modeling information that goes beyond the all-atom structures that PDBs conventionally stored. And they have machine-readable information for linkage to things like cryo-electron density maps and other basic experimental information that from which the models are derived. And so we want to teach VMD to be able to exploit these new structure databases and the linkage that the new MMSIF and PDBX extensions provide. And so this will eventually open up a lot of new opportunities for us to automate incorporation of experimental data into the simulation. So for example, if you get distances from NMR experiments, for example, those distances can become restraints in a molecular dynamics simulation. And we, by being able to read this data, will be able to automate some of those importing of those data into the structure preparation tools like QwikMD. VMD has a lot of tools for taking molecular dynamics simulation and characterizing it in various ways. Some of these are pretty straightforward. Anybody who's done molecular simulations would be aware of, but some of the unusual ones are things like taking all-atom trajectory data and extracting volumetric properties and being able to do things like alignments. Here you see magnesium ion occupancy probability density isosurfaces. So this is sort of an unusual kind of analysis that's easy to do with the built-in tools in VMD. A major area where we've invested both in VMD and in our molecular dynamics program, NAMD, is the development of structure refinement methodology called molecular dynamics flexible fitting, which uses molecular dynamics simulations to augment cryo-electron microscopy density maps with X-ray crystal structures, and using the force field and the knowledge that we have in chemical knowledge in molecular simulations to improve the resolution of the ultimate structure using all these different information modalities. To further augment these tools, we've developed a new plugin for MDFF, which adds tools for interactive GUI-based tools, I should say, for things like rigid body fitting, computing histograms, doing all this sort of piecemeal density map processing operations a person would need to use. We have a density map segmentation tool that's now GPU accelerated, and we can do costly calculations over a molecular dynamics trajectory, such as cross correlations and things like this. And so I'll give you a few examples of those. If you have a bunch of different density maps and you want to compare them, or you want to superimpose regions of different resolution, this is an example where you can use the new masking tool to select different parts of different maps and create a new map of tagged with the different components. And then you can make a visualization like the one on the right, where you can clearly see the regions that have different resolution. The new GPU accelerated density map segmentation tool is in the new version of VMD. It's about 12 to 20 times faster than our best CPU code, and we're continuing to work on this, and our goal is to make this so fast that it is essentially completely interactive, and there would no longer be a need to do things with files. You would be able to do the segmentations so rapidly that you can do it all just by clicking and dragging in the GUI. So this is really a very new feature and very exciting for us. We've never really had, we waited a long time to put segmentation into VMD until we had an approach that would work on the GPU, and so that's pretty exciting stuff. This is used both for, you can use a segmented density map as a way of selecting the different parts of the density map, much like one would select the atoms in the atomic structure with the VMD's atom selection. We plan to extend that selection language to also apply to the density maps and to segmented density maps. This segmentation scheme is also important because we need it for preparing lattice cell simulations. That's a different simulation modality. You can see a lattice representation of a tomogram on the right-hand side. That's one such example. A major part of VMD that we've been working on, I would say from 2011 till now, has been involves parallelizing the analysis of large trajectories. So state-of-the-art molecular dynamics simulations that are studying, large structures like HIV or other large viruses, cellular organelles. I think Abhi will give you some good examples. These can easily produce terabytes of data on today's hardware, and not only for large structures, but I would say some of the state-of-the-art molecular dynamics tools out there today on GPUs, even a moderate-sized structure of only 100,000 atoms, long time scale molecular dynamics simulation of those or with multiple replicas can easily produce tens of terabytes of data. And so we want efficient mechanisms to analyze those. And so we've been parallelizing VMD. Not only does it run on your desktop computer with a GUI and all that, but you can also run VMD with MPI in a purely batch mode with no graphical interface. And that allows you to tear through multi-terabyte trajectories on a supercomputer and compute quantities in parallel. And so one of the early uses for this was for molecular dynamics flexible fitting. We wanted to do cross-correlation quality of fit analysis for large structures, big viruses, and we wanted to do it over 10,000 frames, and we wanted to compute those cross-correlations not just for the entire structure, but we wanted also more spatially localized cross-correlation, and so that drove up the cost of those calculations significantly. We then adapted this code to use the GPU, which I'll talk about in a second, but we ran it on a supercomputer with 2,000 nodes, and this allowed us to do something that if we had used just CPUs and on a single workstation, it would have taken us 5 years with the original code. And running it on 2,000 nodes of a supercomputer with GPUs, we could do it in 19 minutes. And so that's maybe an advanced feature of VMD, but it's something to keep in mind as we go forward and it becomes easier to deploy tools on clouds. We've got a version of VMD that runs on Amazon AWS, and you can do a lot of these same parallel analyses on the cloud as well as on your supercomputer or your desktop machine. We've put a lot of energy into GPU acceleration. VMD is probably the predominantly GPU-accelerated molecular visualization and analysis tool at this point. Just to give you an example, those cross-correlation calculations for a 700,000 atom rabbit hemorrhagic disease virus, doing one of those calculations with Chimera, which was at the time the best tool we knew of, in 2014, that would take about 15 seconds. And over many generations of GPUs and writing our code better and so on, if you do that today on a state-of-the-art Volta GPU, the same thing can be done in 5/100ths of a second with the newest version of VMD. And so, we put a lot of effort into that because we knew that people didn't want to do that calculation on just one frame. We wanted to measure quality of fit over the course of the simulation as the fitting method improved And so this is an example. Another one is for quantum chemistry visualization, large, sort of hybrid simulations that involve both molecular mechanics and quantum chemistry. The state-of-the-art versions of NAMD now link to both ORCA and other quantum chemistry packages. I think we now have support for TerraChem. This allows us to simulate quantum chemical regimes in a molecular mechanics context, much larger than ever before, and so that becomes a significant visualization challenge to show dynamically changing electron orbitals and things like this. But we have support for that in the newest versions of VMD, and it supports not only single GPUs but multiple GPUs and gets tremendous performance out of that. One of the problems we always face is the experimentalists have been so successful in their imaging that they have, over the last 2 decades, made huge progress in the size of atomic detail structures that they've been able to resolve. And particularly now with the impact of cryo-electron microscopy in its heyday, we're really seeing the size and the reach of the structure information is now, the size of very large viruses can now be had at atomic resolution. And so this is pushing molecular simulations in that direction. And so this places a lot of strain on the tool chain, for both preparing and analyzing and visualizing these large complexes. And so we've put a lot of energy into optimizing VMD for, sort of where we think things are going. And to give you an example, one of the things that people want to do with large molecular simulations are to do clustering analyses to determine which structure conformations are salient, which conformations are more stable than others, et cetera. And this is an example of one such calculation that's very I/O intensive. It's both compute intensive and I/O intensive. But if you don't do something about the I/O, it ends up being an I/O bound calculation. And if you have a trajectory that's in the range of 20 to 30 terabytes, that can become a serious problem. So we've put some energy into this. We've been working closely with vendors like NVIDIA, and now there is support for GPU accelerated I/O. So there are ways now for a GPU to directly access files bypassing the host CPUs. So I'm under NDA on various things, so I am limited in what I can say, but I can tell you that, for example, I have an initial version of VMD running on a GPU-accelerated machine that has 16 GPUs and a bunch of SSDs. It has 8 SSDs in it. And with that version of VMD, I can achieve in a single VMD instance, not even running in parallel, I can hit trajectory analysis rates of 20 gigabytes a second. And so you can imagine if you've got 40 terabyte trajectory, that is a lifesaver. We expect to do much, much higher bandwidth than that on network attached storage like Lustre. If you have Lustre like a DDN parallel file system, in the next coming year and a half, 2 years, I expect we'll have performance levels that are a multiple of what I just said on GPU-dense workstations and compute nodes that are common to what people are doing both for molecular simulations, but also for cryo-electron microscopy image refinement and things like Relion. I would expect to benefit from this as well. We're working on making VMD support remote visualization for the purposes as people run these large simulations on remote clusters and supercomputers. It becomes inconvenient to have to transfer large files back and forth, and so for that purpose, we're building remote visualization into VMD itself. So we have a very early prototype in the developmental versions of VMD now, and we expect to get that linked in a more platform-independent way in the coming couple of years. And with that, I just wanted to finish by saying we have a lot of different VMD tutorials. The main VMD tutorial covers basic usage of the tool, how you use atom selections, how to do conventional visualizations, do sort of typical analyses, both interactively and with scripts. And then we have tutorials for particular topics like using QwikMD to prepare and analyze simulations, making images in movies, checking structures, and hybrid simulation visualization for things like CPMD and QMMM, and also force field parameterization with FFTK. And I would like to just thank Klaus Schulten for all the wonderful years I worked with him. And with that, I'll turn it over to Abhi or answer any questions anybody has about my part.
Great. Well, we're transitioning over. We did have one question. A few more details about the storage that you used, your SSD, and this.
So this is a Linux machine that has 8 SSDs installed in it. They're little Samsung SSDs. I think they're about 4 terabyte SSDs each.
Do you know if they were if they're SATA or NVMe?
Those are NVMe. Yeah, and actually, I think I may have underestimated. I think they're 10 terabyte SSDs. I forget. Anyway, they're pretty big. They're big enough for my for my benchmarking purposes. And this is sort of the beginning of this technology. And I would expect. I mean, NVIDIA has it on their website. It's called GPU Direct Storage. You can go read about it. There's a new programming API to make use of it. And, we have, since 2011, we've been developing simulation trajectory file formats that are optimized for solid-state disks. So that helps, too. If you use the right trajectory format and you have everything programmed just the right way, you can get tremendous I/O rates. So even with CPUs, I am able to achieve 10 gigabytes a second, to give you an example. On that same machine with CPUs with exactly the right code, VMD can hit 10 gigabytes a second with the CPUs. That requires about, I think that's VMD running with about 60 or 70 threads, but that's a pretty powerful machine. But that sort of thing is possible in a single box. That's pretty exciting.
Oh, that's fantastic. All right, so we can save other questions. I've got a few here. Well, I'll save them toward the end. But Abhi, are you there? You're ready to jump in with your half?
Yes, I am. So let me then start sharing my screen.
Yep.
Can you hear me?
Yep, sounds good.
And can you see my slides? Okay, good.
Looks good.
So then, let me continue from where John has left, and then let's move forward. So again, I'm Singharoy. I am currently at Arizona State University. I'm an assistant professor here. I have been a Beckman Fellow with Klaus and with John and with the entire colleague there till 2018, after which I moved there in Arizona, and I run my own group here. So we continue using VMD, NAMD, propagating, the strengths of these tools, using them, leveraging them. So what I wanted to talk about today is primarily, as I say, recipes of discovery with VMD. In a way, as a scientist, what do we do with VMD? John was extremely nice in telling us about what are the hardware and software developments, but what do we do with this? What sort of discoveries do we make with that? That would be the focus, the thrust of this part of the talk. With that, then let's move on. The first thing I want to say is not science, but how to popularize science. So VMD is extremely good for making materials to popularize science beyond the community of biophysicists or structural biologists that we are. In fact, this is a part of an NSF-supported project which John helps us with often. So what we are doing is, John mentioned remote visualization, where high school students in the state of Arizona are actually using VMD to create school chapters, which will now be distributed amongst 12 schools using remote visualization. So the idea is pretty simple. You have your high schools, which actually has very, very weak computer background. They have library computers. They have laptops, which are not very, very expensive. Can you connect them to some very strong compute resources? Let's say I'm in Arizona and ASU has good compute resources. Can we use this cloud-based computing, remote visualization, for example, to connect those big computers with the library computers, and then you use VMD and all the interactive tools on VMD to teach the high school students biophysics. This is a project that VMD is literally a core of. You cannot do this without VMD, and so we are working on this. It's a funded project. So in a way, all the beautiful movie making skills that John has shown, in addition to their being important for being great material for manuscripts and eye catchers in conferences, they actually serve a purpose of huge scientific outreach, which is extremely appreciated and kind of awarded by the National Science Foundation, by National Institute of Health. So in a way, VMD serves this extremely good purpose of broadening the impact of science. So exactly, broader impact, that's the word NSF mentions. So VMD is a tool for generating broader impact. Above and beyond, it's being a tool for generating excellent scientific product and for generating excellent discoveries. So this was something I wanted to stress, so that people don't just feel that making that great 3D movie was kind of an exercise in intellectual delight. It was definitely more than that. It was an exercise in broadening, in making sure that science reaches to audience of all ages. And so VMD serves a great purpose there. So before moving to any such discoveries, I wanted to make sure. So the book chapter that you see, or the questions, for example, that you see here, are all made by high school students. And we have 5 such book chapters planned. 2 of them are already made. And in fact, as a high school student chapters, you see a meme. We start with a meme and then we move forward. So yeah, this is one of the great social services that a tool like VMD offers. So do make sure that you kind of utilize VMD in this way. And I think there are high payoffs for that. Thereafter, now let's move on to then the discoveries with VMD. So the first thing is, of course, we are very, very interested in studying big systems, but at a molecular level. So we used to call this, Klaus used to call this, and we carry this forward, we call this structural systems biology. So what is structural systems biology? You have a bunch of proteins that, for example, you obtain by solving their crystal structures or by doing the electron microscopic structures. But those are just individual proteins, single protein structures. Biology is not about individual proteins. Biology is in fact about how these proteins work with each other, how they coordinate, how they combine with each other. So then let me kind of give you a workflow of what we call structural systems biology, and then take you to places where VMD comes in. So what do we do in structural systems biology? We take a bunch of proteins. Then there's a big number of experimental data that is known in terms of how these proteins are mutually arranged, how they are placed next to each other, how do they interact with each other. John, of course, mentioned Cray-EM, and we will see a bit more about Cray-EM, but there are 10 other kind of data modalities that can be folded in to understand the organization of these proteins. It can be AFM, it can be mass spec, and there are different ways in which you integrate them. I will not be able to get into the gory details, but the idea is that you can actually start integrating experimental data at different resolutions. Cray-EM and X-ray crystallography is definitely very high resolution and getting to be high resolution. But then there are other data modalities that are low resolution for the purposes that they serve. You really need low resolution data to understand organization at a larger level. So fine, you have your protein structures. Somehow, and I'll gradually tell you what that somehow is, to get all of these proteins together, and you can do that on VM. This is where we interface with our favorite tool, NAMD, Nanoscale Molecular Dynamics, where we can actually simulate these objects. For nowadays, actually, we can simulate them from microseconds and extract millisecond scale information. We will comment on this a bit. So we do our rates. From there, we compute molecular rates at which these molecules move. Thereafter, but again, you see all the visualizations are on VMD. So VMD serves this purpose of visualizing not only stationary structures, but also mobile, movable quantities. Thereafter, you do your maths. This is something that's not within the realm of VMD or even NAMD. You have to do your maths, where you plug in the rates that are obtained from these molecular dynamic simulations, and from there you actually end up predicting cell scale quantities. You end up predicting phenotypes. You end up predicting something that you wouldn't have imagined can in fact arise from molecular details, but you end up doing that. So this is what is beginning. So as John had mentioned, people are going into bigger systems. They're going into extracting atomic scale knowledge of large systems. But what would you do with that? Here is one example of what you can do with it. You can actually model really, really huge systems, and then using your skills of VMD, your skills of NAMD, and of course your skills of physics, that's very, very important, you can end up predicting cell scale or even organism scale properties. What we ended up predicting here is that the particular organism, bacteria to which this organelle belongs to, is a low light adapted bacteria. It can only work under light intensities that is 1/10 the amount of light that is present under regular in a regular day. That means that this particular bacteria can live under the ocean. You cannot bring it out, that this bacteria will die otherwise. But that's kind of a prediction that's made using molecular details. That's the uniqueness of it. So you can actually start asking the question, how does atoms interact with each other to become molecules? How does molecule interact with each other to become proteins? How does protein interact with each other to actually give you this real biological properties? So as a theme, this is what I think a bunch of scientists, a cadre of scientists who, starting from Klaus Schulten's lab and beyond, we're following this theme that how can we extract system level properties from molecular details? Systems biology already existed. We bring in structure into systems biology, and VMD is the most essential tool to do that. So this is the kind of the science overview, if I may. Now let's dive in. How is VMD helpful? So the first thing, as John mentioned, is we can tie in experimental data. So that we can, of course, tie in X-ray crystallographic data. We have a tool called MDFF, where which is not a phasing tool, but it's a real space refinement tool. The density, the electron density gets morphed during the simulations, and towards this morphed electron densities, the side chains of a protein gets fit. So it's a side chain refinement tool, if I may. It works for low resolution X-ray data. We have tested this for 1.5 to 7 angstroms, up to 7 angstroms X-ray resolution, very low resolution X-ray, which people normally just throw it out. But we in fact worked with pretty low resolution data and showed that we can get structures commensurate to high resolution structure from low resolution data. This can be, so these simulations can be run on NAMD and can be set up on VMD. I'll show that to you in a bit. The next thing is when you have large scale organizational changes, you can, of course, work with MDFF, which is a way of fitting atomic models into Cray-EM data. Nowadays, of course, Cray-EM and X-ray resolutions are matching. The lower end of Cryo-EM is, the lower end of X-ray crystallography is overlapping with the higher end of Cryo-EM. So there's a lot of overlap between MDFF and XMDFF in terms of structure refinement. And after that, you can, of course, the beauty of VMD and NAMD is you can do this in real membrane conditions. You can do refinement in literally in silico in situ, if I may. So a computational in situ kind of an environment can be created, which possibly cannot be done in any other structure refinement tool today. You can actually do these refinements in real membranes, in real surroundings, as much as real as we can get. We can actually create a membrane and refine inside that. And then, of course, once you make each one of these proteins in their own atomistic forms, then you plug them into a big membrane. How to plug them into a bigger membrane is the next slide. But let me tell you what is there on VMD that allows you to do this. So you have this MDFF plugin on VMD. It's an MDFF visualizer. I think if you go to VMD, then if you go to modeling, then you will find MDFF setup. Here, of course, you can just load your PDB file. You can load your density file. There are a number of other parameters that you can leave for default. You can set out your simulations or refinements in vacuum, under periodic boundary conditions, under implicit solvent if you have to. You can do XMDFF, where of course, in addition to the structure, you'll also have to provide your intensity data, and any standard format intensity data will be accepted. That's not a problem. And in order to create, for example, the membrane, that you have to, we actually have a charm GUI. There's a web-based facility. You can go there and you can do exactly the same thing, and you can now also add the membranes there. Membranes haven't yet been added to VMD, but you can add the membranes on charm GUI. It's a very, very user-friendly interface. You plug in the same kind of user input, and you'll be able to create all the necessary input files, which you will run with NAMD for running MDFF or for running XMDFF. And the beauty is you can do that, I repeat, in any sort of environment, explicit solvents, implicit solvents, and membranes in the presence of ions. I think that's very unique. You make the best use of biophysics as well as structure refinement. Okay, that's one example. Now, of course, you have all these proteins. So you have individually refined each one of these proteins, let's say the green protein and the purple protein and the brown protein. Now you want to organize them in this particular shape. This, of course, takes, fortunately or unfortunately, this needs some expertise in scripting with VMD. So I will not be able to give you details on how to do it, but I will be able to forward you to this link here, which actually has a lot of TCL scripts that is used to organize individual proteins into larger scaffolds. So there are more intelligent way of, I would say, less scripting, more interactive ways of doing that. It's amenable to an advanced user. I should say that caveat. There's something called instancing, multiple instancing on VMD. Might be John can comment on that after I am done talking, where these sort of things can be done in a bit more interactive way. But I would suggest that a good VMD user is a person who knows how to script in TCL. So that's in that way VMD can become like your pet in a way. As Klaus used to say, it's like a car. When you start driving it, first of all, you enjoy it, and then you gradually can get into the nooks and crannies of it. So I think VMD is an amazing car to drive. So if you can learn TCL and look into some of the scripts, then you'll be able to figure out how to organize these proteins into very, very complicated shapes. So how to deal with shapes in VMD, I would say, is something you'd need TCL scripting for. Go through the scripts if you have the time. This is kind of, as we say, a library of all possible things that you can do with VMD using this TCL scripts. There are at least 40 to 50 scripts of different purposes. If you go through the scripts, you understand them, then you'll learn a lot of them. VMD also has its own script library. You can look for VMD TCL script library online, and you'll find that. Between those two scripting languages, you'll be able to organize these big scaffolds. Okay, now coming back to chromatophore. So once you have the scaffold made, for example, then you want to build a membrane around the scaffold. The first thing you do is you build a separate lipid vesicle. How to do that is also there in this script. Then you overlay them, and then you exclude all the atoms that is kind of conflicting between the positions of your proteins and your lipids. And then you do a little spherical boundary condition simulation. And after that, you make this big hexagonal box. And in fact, in NAMD, we can actually do hexagonal periodic boundary conditions to make this huge system. This is a 106 million particle system. And then you start simulating this. And long and behold, you see your simulation is unstable. Really, really bad. You see that there are holes coming out. There are water that is escaping from all sides. This is not a stable simulation. So even after you're doing a lot of things with VMD, we follow that with NAMD, you get unstable systems. Especially this sort of problem is very, very common if you have a container that looks like a vesicle or any other shape, and primarily if it's a lipid-rich container. If it's a protein-only container, it's still doable. It's still manageable. But if it's a lipid-rich container, you see these big holes coming out. So how do you get over issues like that on VMD? So my friend Juan, together with John, they created this tool, if I may. It's called a biomolecular container analysis. What this does is it actually gives you a very up close and personal idea of how molecular surfaces should look like. Based on that, what the first thing you'll be able to tell is are you inside the container? Are you outside the container, or are you within the container? And think about biological containers. They are thick containers. They are not like a single film. Biological containers are often 30 to 40 angstrom thick and uneven. So you literally need to find out molecular shapes to actually understand what is the density of solvent inside and outside. If you see the lower right, the simple sphere example, it's very simple to fill that container because the density is quite uniformly spread in the space. There is no spatial homogeneity in density. So it's very simple to solvate, add water to the systems or add membranes around it. However, if you move to the left, you see the molecular shape has become extremely contorted. It's a very, very difficult computational problem to actually count the number of waters inside this container so the density of water remains 1. But with these tools, you can actually end up doing it because you now know an exact idea of what the molecular surface of a virus looks like. So with that then, so again, this tool is available on VMD. Make the most of it for solvating systems. So after doing a simulation like this, and it was, using tools like this, could we actually stabilize this big organelles to actually start envisioning to do this system structural biology. But it's very, very important that we make sure that the molecular shapes are given due credit when we do density calculations to fill in water. For a normal protein, for smaller systems, it's okay, you can live with it. But for these sort of systems, for these really large-scale investigations, you need very proper ideas of molecular shapes, and your biological containers would actually give you a very, very good idea of what the shape is. Again, go back to the TCL script shown in the last slide. You'll be able to have some idea about how they are. So how these slides, how these things work at scale. You need to get your hands dirty a bit to understand. But okay, so this is the next option, biomolecular container analysis. So I showed you how to do MDFF, biomolecular container. Let's move on. VMD is very, very nice for computing electrostatic potential properties. What I show you is how the electrostatic potential inside a particular cell organelle is changing with salt conditions. It's very, very important to note that at high salt conditions, screening effect comes in, and the electrostatic potential, so the red color here is negative electrostatic potential. At very low salt conditions, they're more spread, and at high salt conditions, they kind of become, very tight. There is a scientific implication of this in the field of molecular recognition, but we won't get into that. For the time being, the two take-home messages are that, first of all, there's a visualization of electrostatic properties that can be done with VMD. And the next thing is you can set up this sort of simulations on VMD. So I'll quickly jump there. No worries about this movie. So there is an APBS plugin on VMD. What you can do is you can actually load your PDB file. It will convert. The first thing, of course, you have to do is you'll have to load your PDB files. It will be for small systems, for system sizes up to 99,999 atoms. You'll be able to generate a PQR file, which is needed for APBS to run. And then you'll be able to tinker with all the settings. And finally, you'll be able to get an electrostatic potential like this, which often, of course, is a very, very important property, a biophysical property. But in addition, these potentials actually are input to subsequent coarse-grained simulations called Brownian dynamic simulations, which is available with NAMD. It's a tool called Atom Resolved Brownian Dynamics. It's available on the TCVG website. But the input of that can be prepared on VMD using this VMD's APBS plugin. Okay, so at least I showed you 3 examples, which makes today this large-scale simulations a reality. The presence of the MDFF tools and MDFF support, the presence of the biomolecular container analysis, and now the presence of this electrostatic analysis tools. Now let's quickly switch gears. Let's go to electron microscopy. So EM Data Bank had actually, every year, they announce a modeling challenge, and they give a number of challenge densities, and you're supposed to come up with the best possible structures, and then people compete to find out which structure has scored well. So we competed in all 4 categories this year, like last year. So we have been like the MDFF team, if we may, is a regular visitor attendee of this challenge. So I want to focus on a particular problem. This problem deals with protein and ligands working kind of stuck together. So you have to resolve a density, a part of which actually has proteins and the other part of which has ligands. So how do you deal with a problem like this? Because the biggest problem is the binding pockets about your ligands are actually very, very, you really don't know what kind of interactions are there. Often the resolutions of these densities doesn't give you much about very specific protein ligand interactions, but you know that these are extremely specific interactions. That's why these ligands are there. So first of all, how do you solve this sort of a problem? And then how can VMD help you solve this problem? So then we came up with a workflow of how to solve this kind of problems. So there are 3 different layers to this workflow. First, we take some sort of real space refinement. Let's say it comes out of Phoenix, which gives us an initial guess of what the structure is going to look like. Then we use Schrodinger tools. Schrodinger, unlike us, we are open source. Schrodinger, you'll have to pay that. So we had to pay for Schrodinger tools. We bought their licenses, and then there's a tool called glide docking. So we did glide docking on Phoenix refined structures to get our initial model. Now then, depending upon the resolution of the map, if it's very high resolution, then we know that the map has all sort of information. We go on the left-hand side of the fork, we do a simple MDFF calculation. If it's a low resolution data, then we did something that we call an accelerated MDFF calculation, where we couple something called molecular dynamics cold-vars, specifically quad num as a collective variable. So coordination number is a collective variable that actually defines binding pockets inside NAMD. So we combine coordination number or quad num with MDFF to come up with the next level of refinement. Finally, what we did was NAMD right now actually has an interface which can which can spatially decompose a system. A part of your system can be one force field, and the other part of your system can be a different force field, or can even be a QMMM calculation. So then what we did was we selected our drug part, and we either did machine learning on that part, on-the-fly machine learning, or on-the-fly QMMM. And the other part, the protein that was surrounding it, we followed that with normal charm. And then finally, we did some model quality analysis. This was the workflow that we actually had to go through to kind of appear in that competition. Now that it is done, it's there in VMD. You go to QwikMD, and QwikMD actually can allow you to do SMD, which is steered molecular dynamics, or in other words, accelerated MDFF, if I in the same kind of spirit of things, MDFF, QMMM, everything can be made using this GUI actually. Here is a closer view of what are the QM options that are present there. And of course, you can look into what kind of code that you want to choose. If you see at the lower end, I have the QM software. My mouse is not showing up. Otherwise, I would have pointed that you see the word ORCA written there. That can be a Gaussian. That can be some other quantum code. So this hybrid, I would say this is like the best possible hybrid simulation I've ever seen in my life, where you combine 3 layers of hybridization: a QMMM, a refinement, and a steered molecular dynamics, which means some sort of a statistical sampling. All the 3 happening together. And you can set up all this on VMD, on QwikMD, in fact. So this is very, very powerful. Okay, I think I am running out of time. So I had one more thing to show. Okay, let me just take 2 or 3 minutes. And after that, of course, you can, as John was telling, you can start analyzing things on VMD, how good your fitting is. So you can actually do this VMD's timeline, which actually tells you on the fly how cross correlations are done. So VMD's timeline plugin is a very, very good support for post-simulation analysis. It can be MDFF, can be MD simulations. You can see all the possible things it can analyze for you. It can analyze secondary structure. It can analyze for you, let's see, hydrogen bonds, salt bridges, RMSD, RMSF. SASA is solvent accessible surface area. So it does a lot of analysis, not necessarily on the fly. Work is being done to make this on the fly. But as a post hoc, you can load your trajectory on VMD and use the timeline tools to kind of get a very good idea is how your system is changing as a function of time, how various spatial properties of your system is changing as a function of time. One last slide. Of course, we did quite well after putting so much firepower. We did quite well in the competition. Green means good to acceptable. Red means where MDFF team was. We were pretty much on the higher end, on the right-hand side of this plot. Good. So then one last thing I want to show is we can do biochemistry with VMD. We can actually do molecular mutations. So on the left-hand side, I show you a movie of a biological motor in action. And so again, we have some sort of residues that we can identify in these movies, and we want to know if we mutate these residues, what's going to happen to this particular to this particular system. And so here is our Mutator tool. It's again in the modeling part. You can actually predict mutations, and finally, of course, if you have experimental colleagues, they can go ahead and then test those mutations and see how they affect the activity. But this is again one last plugin that I wanted to share. VMD Mutator is very, very good in identifying residues, doing an alanine scan, a computational alanine scan, which is also available on Rosetta. But this we can do in we can find very accurate energies, and then we can make predictions that can be subsequently predictions of binding affinities that can be subsequently experimentally validated. Good. So with that, I think I have given you a list of all the possible VMD plugins, and I end my my talk here.
Great. Thank you very much. I got a few questions. If anybody else has questions, you can send them to me by chat, or I can also unmute you possibly, and we could ask just one of the questions and maybe Abhi and John, you can kind of figure out who this best addresses, is that Cryo-EM is steaming ahead. You know, the single particle reconstructions but cryotomography is really kind of lagging. Like, there are people who are looking at determining structures in cells or in like the context of, and to do this, they need to have some sort of automated way to recognize compartments and particles and do segmentation. You talked a bit about the containerization and the segmentation in VMD. Is that something that's like on your radar or sort of in the workflow for VMD right now?
So it's definitely a forward-looking area. So when we started our current funding period, we did sort of a survey of where things were going, and we talked to a lot of our close colleagues. And some of our colleagues in Cryo-EM were actually telling us that they were making a transition themselves from Cryo-EM towards cryo-electron tomography, and that they felt, and some of their colleagues felt, that tomography was going to be approaching the resolution that single particle reconstruction gets now with Cryo-EM in a certain number of years. Now, that's probably I'm sure that's a hotly debated thing, and I'm not an expert on those things, but that's the advice we got. And so one of the things they directed us to think about was, they posed the challenge. So now imagine you can get a tomogram that's near atomic resolution. Maybe it's 4 angstroms or something like this. Maybe it's 3.5. That's what they were talking about. If you have something the scale of an entire cell, now you want to automatically label all of the contents of that cell by recognizing its constituent particles and things like this. An example would be, if you have ribosomes all throughout that cell, you want to label all those ribosomes. You want to identify what these different components are, all the microtubules. Today there are tools that do some of these things sort of in a very human-directed way. You can basically drive a imaging tool to segment and label these different things and do atomic structure fitting to the tomogram. But this is a very arduous process, right? And so there's a vision that with the advent of GPU accelerated algorithms and opportunities in imaging that are coming in the near future, we should be able to do some of this as an automated campaign where we let the computer, maybe it's not a desktop computer, maybe it's a cloud. We use parallel algorithms on multiple GPUs, and we can do things like a correlation field for all the different things that are in one of these tomograms, and have the computer do most of the legwork. And that would relieve the experimentalist from having to manually drive user interfaces to pick all these little things and do individual cross correlations sort of the hard way. This is a tremendous computational challenge, but it does seem, if I were to predict, I would say in the next 5 years, we're going to see yet another quadrupling in the speed of GPU hardware, at least, maybe more. And for certain calculations, like some of these, what should I say? I mean, definitely, I would say different kinds of masking operations and things where we can exploit. GPUs have special hardware now, these so-called tensor cores that reach into the hundreds of teraflops per second. If you can turn your calculations into things that are amenable to that particular type of hardware, you can achieve performance that is almost unthinkable relative to what we've had up to now. And if we see hardware continue to evolve, which I think we will, there will be greater diversity there. I know, for example, the tensor cores on the first generation Voltas are, they have a tremendous performance level already in Turing. They've added more features that are maybe actually better suited to image processing because they work on half precision quantities. So these are 16-bit floating point numbers. That's a lot closer to what we see in the tomogram. So you'd be able to get a better performance with smaller data like a tomogram. And these different types of, sort of filtering and correlation algorithms and things like this are well suited to that hardware. And I think if they improve the generality of the programming just a little bit, we'll be able to get all of a sudden it'll be just the shoe will drop and there will be another factor of 10 even beyond the best performance a GPU has ever seen, because you'll overnight be able to use it versus maybe right now there are various things that prevent you from exploiting it to its full potential. They're just beginning. For example, now we have math libraries that are able to exploit that special hardware in very ingenious ways that nobody really thought about. It's taken a couple of years for that to happen, but now we have linear algebra libraries that exploit tensor cores and mixed precision to achieve performance levels that are several times faster. And they don't fully use that tensor core hardware to its full potential. They have to make various compromises because right now that hardware is very limited and inflexible. But already you see performance levels that are maybe 4 times what it would have been with the general purpose floating point units. And so if we can exploit some of that hardware and we design algorithms and software, we'll be able to do some pretty amazing things just with a single GPU. And then the hardware is going in a direction where it's now easier to exploit several GPUs concurrently, even in a desktop machine. And the memory capacities of state-of-the-art GPUs now reaching 40 to 50 gigabytes on a single GPU. It won't be very long before a lot of the things are possible with, some of the state-of-the-art tomograms are really huge. I think I have a test case I use for exercising VMD. I have a tomogram that by itself, one tomogram is 60 gigabytes. So we're now seeing the top-end GPUs are reaching into that territory. As long as they keep pace, and I think they will, that's a great opportunity to apply them.
Yeah, a sort of related question from Jared Smith. What are the relevant tool chains for this and, for example, the GPU direct I/O stuff? What are the relevant tool chains you're using?
So GPU direct I/O, this is, NVIDIA calls it GPU direct storage, and they have another thing they call Magnum storage or Magnum I/O as I think the feature that they refer to on the DGX workstations that they make. Anyway, so that goes with CUDA. It basically adds features to CUDA that allow you to read and write files straight into a GPU memory system, bypassing the host. And if the workstation or compute node that you're using has the right combination of hardware, this can give you a tremendous performance boost versus what you would have seen with a CPU. And, while as I say, there are lots of things that I know that I can't yet share. It isn't very long, I think, before a lot of this stuff will become public and you'll hear a lot of noise made about it. As you can imagine, there are a lot of deep learning workflows, AI workflows that are very data intensive, and so I/O is critically important to them. And so those of us here in the computational chemistry and molecular modeling community are going to get a sort of a free ride. And I've been very fortunate to have an inside track there to get access to this stuff in its early implementation, while it's still in development, and to give them feedback about the things that are important for us, for our community. But I think NVIDIA is going to announce a lot about this at their GPU Technology Conference in March. So it's, I think March 22nd is when you're going to start hearing a lot of news about that. They have an article posted, I think, already last year describing the overall idea and how it works and what applications they have used it for themselves. They have a programming NVIDIA blog where they describe that GPU I/O. But anybody who's interested, I am happy to answer questions offline, and I'll share any information I'm allowed to. And I can put people in contact with, NVIDIA or whomever if they're software developers themselves.
Got two more questions. One from Pete. Do you think that the cloud I/O will be able to keep up with the improvements in the compute hardware?
That's an interesting question. I think that there's a lot of action right now in this, what do they call it, converged network space. This is where you're using InfiniBand type hardware to do everything. It acts as a storage system. It acts as a low latency compute fabric for sending messages, and it acts as your Ethernet, as it were, to the outside world. I think the kicker is it really depends on the hardware provider. So, for example, Amazon has historically been an Ethernet-centric business. So they didn't have super low latency InfiniBand hardware historically. They were an Ethernet shop, and that had to do with the convenience of their virtualization. I think that it's difficult to say how this performance will behave on something like Ethernet. I would say I'd worry that Ethernet might not be able to keep up. But I can tell you for sure that the different hardware companies are currently collaborating, so that I have seen and heard of things on InfiniBand type hardware where you're getting basically the same speed or better. In some cases, significantly better speeds are possible due to larger storage systems. And I think it then is a question of what that cloud provider has. So AWS will be a little bit different from Microsoft Azure and the other options that are out there. I think you'll have to evaluate those on a case-by-case basis. But I would expect that certain cloud providers will probably end up specializing in I/O-heavy apps or things that need network-based storage, but also have significant bandwidth requirements. Another thing is that I expect also the early version of this on the GPUs. Keep in mind, this is like the version 0 or something is what we're talking about now, and I can get 20 gigabytes a second without too much effort. I guarantee you that over time it will get better and it'll get easier, and it'll be more tolerant. So it could be that while right now I wouldn't expect an Ethernet-based network storage system to be competitive with direct access to NVMEs or direct luster over InfiniBand, maybe in a year's time, that'll be possible too. I just don't know yet. It's very early days, so we'll just have to see where that goes.
Yeah, it's interesting. So another question I had, kind of unrelated, but still hardware-centric, is that NVIDIA has made this 3D vision that structural biologists had relied on for years. It's this shuttered glasses with a high-frequency monitor, and they're not making it anymore. So they don't make the glasses, or they're not supporting it past, I think the last driver came out in April, and after that it's going to be gone. So this leaves structural biologists without their sort of 3D visual tools to look at density that's in this range where cryo-EM kind of lives, this sort of 3 angstrom, 4 angstrom, 5 angstrom area. I'm wondering if tools like like VR or augmented reality tools would be able to sort of fill in there, and if that's something that VMD would be sort of primed to do, or if it's even something that maybe you've heard people raise before.
So, well, I'm keenly aware of this problem. Our laboratory operated stereoscopic projection systems for the entire time period I've been working here, and it was that way even the 10 years prior to my arrival. You may know that, the shutter glasses started with Evans and Sutherland in the early 90s and late 80s time frame. I actually know the son of the inventor of stereo shutter glasses. Anyway, so they invented that technology. It's been in Silicon Graphics workstations. VMD already had support for shutter glasses in '98 when I started working on it. And we've relied on that, and so have all crystallographers for that entire time frame. And yet a couple of things have happened. You've probably also witnessed there's been sort of a sea change in the way laptops are constructed. Today, laptops are now built with the same display format as HD TVs, and it's sort of the crushing pressure of the economies of scale has pushed all computer equipment in the direction of adopting whatever it is that televisions are using, and they use the same displays and display panels and everything for all of this stuff. And the difficulty is, I'll be honest, what really has happened here is shutter glasses have died off simply because high frame rate displays that are capable of 120 or higher hertz have become rarer than hen's teeth, as they say. And that was precipitated by the death of stereoscopic TV, sort of failing to become a major player in the mainstream television viewing. And so what's happened is you can't buy stereo TVs anymore, and not just active stereo like you're describing with shutter glasses, but you can't even really buy passive stereo displays that have polarizers or alternating scan-line polarization, things like this. There are a bunch of other technologies that are alternatives to shutter glasses, but I'll be honest, all of them are basically gone. And so what has happened is the commoditization of VR headsets, led the television manufacturers to decide that they didn't want to try and compete with that. They were never going to have a 6 degree of freedom virtual environment viewing experience like a headset would have. But this has really become a problem because, as you're probably aware, when we as scientists look at a stereoscopic display, oftentimes we want to have a meeting with several people, and we want to look at a large TV or a projector or something like this, and the shutter glasses allowed a group of people to interact with stereoscopic visualization very easily. And the problem with headsets is that they are isolating. When you're wearing a VR headset, you can't see your colleagues and they can't see you. And this is sort of an unmet technological barrier to using those as a meeting tool. So you can't have a group meeting and a real scientific discussion very easily with the headsets a priori. Headsets are still in a very infantile state with regard to operating system software and how they're integrated into the windowing system. They're really considered as an outlier. They're not even given first-class display treatment. They don't integrate with the Windows shell. Platforms like macOS don't really have support for headsets at all yet. So we have a bit of a Wild West going on. I will say that for an individual scientist sitting at their desk, I think a headset is an adequate replacement, and in many ways it's a superior replacement for stereoscopic shutter glasses. Certainly they have higher visual fidelity with no ghosting, things like this. Particularly better quality headsets that have glass lenses and good optics can have razor sharp images, unlike anything we've ever seen with shutter glasses or even passive stereo. And so there is an opportunity there for us to have actually a better outcome than what we had ever had before. But the missing link really, in a lot of cases, is this assumption that people work completely in an isolated fashion. That's not really how we work. A lot of the work we do is collaborative, and so one of the challenges we have to overcome And I don't even think this is the job of a tool like VMD necessarily, but we need to have a way to network a large number of headset wearing people together into the same virtual environment so that they can interact with each other in a natural way, and we undo the damage that that is done by the isolation that the current generation of headsets enforce. There is some, if you look in the technology field, they refer to, they don't just talk about VR or virtual reality or augmented reality, they call it XR. There's sort of a continuum from VR headsets as we have them now that are a commodity toward what they hope to have with augmented reality headsets, where one day you'll wear something that looks a little bit more like funny sunglasses or Google Glass maybe, and you'll have an immersive display that's easy to use, doesn't create focus-induced headaches, things like this, and that would let you also see your colleagues. And by having a shared space, a projection that's adapted to the viewer's location in a room, it would be more like what we had when VMD was first written. VMD originally ran in the cave, which is a 6-wall projected virtual reality environment where the main viewer has a 3D tracking device, and as they walk around in the room, the projection follows their view. Well, of course, they've evolved since then, and they now have caves where multiple viewers can be in the same space. And we can do that with a cave with wired tracking equipment and so on, but what we need is to be able to do that with commodity hardware, with these headsets, and have people work in the same space and to have more of an augmented reality type display so that you can see the virtual objects, the molecules, in the space between us as we're talking, and we can point at something, and when one of us points at it, the other sees our finger pointing at it, and it is the molecule, and when they point at it, I'm seeing the same thing, but from my perspective, that they are. And once we get to that point, then I think we'll have really gotten back to where we were, but you can imagine there are a bunch of technical barriers to achieving this that are as yet unmet. So I do think that VR headsets are going to take the place of stereo TVs, shutter glasses, all of that equipment. All of that is going to be a relic of history very shortly, and I think that's why you're seeing it being abandoned by the hardware vendors. But the problem is we haven't really replaced it in the way that it was actually used, especially for meetings. And so that's where we've got a lot of work cut out for us, and we have to walk before we run. But I've been keenly chasing after opportunities to incorporate multi-user VR visualization into VMD, and I can tell you it's non-trivial, but it's what I'm hoping to achieve by various routes.
Well, that's exciting. Yeah, it's job security. We have lots of important things to do. Well, great. Well, I think we should wrap it up. But Abhi and John, thank you very much. A lot of exciting stuff, man. There's a ton of VMD plugins. I'm gonna just go geek out on VMD plugins for a while, I think, after this.
Great. Well, as I said, please don't be bashful. I would advocate anybody who has questions, join the VMDL mailing list. Visit our website. We have tons of tutorial material. I think between VMD and NAMD, we have over 50 tutorials. And, if you have questions, ask on the mailing list. We have lots of friendly people that help. And so don't be bashful. Please get in touch with us with any questions.
Great. Well, thank you very much. And thank you, everyone, for joining. Take care. Bye.
Thanks.
Bye.